Thank you, Claire, for the introduction, for organising and for the opportunity to speak today. So I'm talking to you from the Geoffrey Pope building, and that's home to the MRC Centre for Medical Mycology. It's situated on the very beautiful Streatham campus at the University of Exeter. And as Claire mentioned, I'm a fungal geneticist by training, and I'm an infection biologist by research. And I have an interest in fungal species that thrive in the natural environment, but that sometimes cause very serious human diseases. And the important role that many of these types of fungi play in the annual turnover of organic matter in the environment is depicted by the difference between these two images of the campus. So fungi are all around us, and every breath that we take is full of fungal spores. In fact, with the obvious exception of diseases which are caused by the commensal yeasts that colonise our guts and skin, the overwhelming majority of fungal diseases that can kill a human being are acquired via the inhalation of a fungal particle. When we think about the global burdens of fatal respiratory infection, there are a number of important factors to consider. One is how frequently such diseases occur, and another is how many of those infected will die from their disease. The number of deaths occurring is the product of these two factors, and these metrics are often used to measure how well we're doing with tackling particular diseases. So in 2020, over 100 million SARS-CoV-2 infections were reported, leading to 1.8 million deaths, a case fatality rate of 1.8%. This is better than the management scenario of TB that caused a similar number of deaths that year, but affected far fewer people. And it's worse than malaria, where many more people were infected, but far fewer died. Deaths from invasive fungal infections also reach the millions in number annually, but on average they kill 50% of those infected. And many of those deaths occur despite antifungal treatment, and mortality rates for drug-resistant disease can approach that of Ebola, for which there's no known cure. So the MRC Centre for Medical Mycology was established in 2009 to tackle these challenges by building capacity in basic and clinical mycology expertise. And research in the centre now covers 5 research themes that collectively seek a better understanding of fungal diseases and to drive the development of diagnostics and antifungal therapies that can improve their prognoses. Around the globe, the common mould Aspergillus fumigatus accounts for a significant proportion of fungal respiratory disease. So this fungus is a soil-dwelling mould, it sporulates abundantly and it grows rapidly at 37 degrees. And the fungus propagates by releasing these melanised spores, which are extruded from a vertically growing specialised fungal cell. And they're dispersed into the air, and they're designed to withstand stress and to fend off predators in the environment until a new moist and carbon-rich environment is encountered. And at that point, each of these spores will undergo a new programme of growth involving swelling, germination, and rapidly branching growth of these elongating cells called hyphae that you're watching in this movie right here. So the extending hyphal tips of the colony secrete a cocktail of enzymes and toxins that's highly damaging to human lung cells. And this facilitates their penetration into the tissues and the blood cells of the host. And this growth programme is similar in liquid medium in the lab and in the tissues of immunosuppressed mice and human hosts. Aspergillus species cause a range of fungal lung diseases in various clinical settings that can be rapidly fatal, chronic, or allergic in nature, and now include viral pneumonias as risk factors for acute disease. Across this spectrum of invasive and chronic disease, frontline therapy, including prophylaxis in immunocompromised hosts, is reliant upon a single orally bioavailable drug series, the azoles. However, in recent years, the dual use of azoles as medicinal and agricultural fungicides has accelerated the emergence of clinical azole resistance. And we know this from the spatio-temporal trends in reporting of azole resistance across the globe, depicted here in this video in work by Matt Fisher and colleagues at the MRC Centre for Global Infectious Disease Analysis, who are leading the global effort to track and trace the ecological and clinical drivers of this problem. And this problem has a measurable human cost, as evidenced here in clinical data from Paul Verwey and colleagues in the Netherlands, that revealed voriconazole resistance to be associated with an excess mortality of 25% at day 90 in patients with invasive aspergillosis. For aspergillosis, the diminishing utility of the azole drugs is a serious health concern because useful alternatives are few and they are flawed. So agents that target the hyphal cell wall, such as the echinocandins, are able to lyse the growing fungal tips, but not often able to kill them. And in contrast, agents that target the peripheral membrane of the fungal cell can rapidly kill the fungus, but they're also toxic to mammalian cells. And currently neither of these classes of drug are available in oral formulations. There are a handful of new agents progressing through phase 2 and 3 clinical trials, but the discovery pipeline is sparse and it's underfinanced. And so we will require sustained activity in this area to offset the problem of antifungal resistance that will not be going away. In bacteria, the evolution and dissemination of antibiotic resistance has already outpaced the development of new drugs. And this has led to a lot of interest in other approaches. Recent surveys indicate that screens for directly acting small molecule growth inhibitors now account for less than 50% of all new discovery programs, and more than a quarter of new efforts seek agents which involve antibodies or immunomodulators or anti-virulence drugs. And anti-virulence approaches focus on disarming the pathogen by neutralising their virulence factors. And this is an approach that might be useful to circumvent the severe selective pressures imposed by blocking essential cellular processes. And because the screens that are used to identify anti-virulence drugs are bespoke and cell-based, they offer new opportunities to screen existing chemical libraries. Also, the use of these sorts of agents as adjuvant treatments alongside existing antibiotics has the potential to promote more judicious use of increasing antibiotics and potentially to increase their efficacy. So in this talk, I'm going to draw upon my own research as an example of how we can use an anti-virulence approach to drive the discovery of new antifungal drug targets and small molecules that act upon them. And although I'm going to focus upon Aspergillus fumigatus, the underlying principles and research methods apply to all of the organisms and diseases that CMM researchers here are working on. So starting point for any antivirulence strategy is to ask, well, what are the pathogen factors that drive disease in the human host? And although there are many dozens of Aspergillus species in our natural environment, Aspergillus fumigatus is one of only a handful that cause human disease. Now, genome sequencing efforts have yet to reveal a single genetic trait that promoted this evolutionary leap to pathogenicity in Aspergillus fumigatus. However, it's the most thermotolerant species that seem to be problematic in humans. And at the beginning of our research programme, we reasoned that it would be the ability of Aspergillus fumigatus to adapt to the differing environments of the natural environment and the host environment that would be an important driver of the disease process. So our idea was that the presence of fungal genes in a fungal genome would not be the sole determinant of pathogenicity in that species, but also the timely expression of those genes in accordance with the extracellular environment that the fungus experiences. The making of a cake that can't be achieved simply by throwing all of the ingredients into a bowl is a useful analogy here. So fungal cells continually gather and process information about their surroundings to guide the production of cell wall components, enzymes needed to degrade complex nutrients, and toxins needed to fend off predators. And genes are the fundamental units of DNA that drive a functional response to an extracellular stimulus, often via the action of regulatory proteins called transcription factors that bind to the DNA in the neighbourhood of genes that have to be expressed in order for the fungus to adapt. And this drives the production of a chemical template called a transcript that informs the production of a specific protein. The quantification of these transcripts, either individually or en masse across the genome, is a sensitive measure of what the fungus is seeing in its immediate environment. And interfering with the accuracy of this information processing is a powerful means of preventing fungal growth. So what follows are some examples of what we have learned via various interventions into this process, culminating with some current work where we ask the fungus itself to tell us which drugs will best interfere with its ability to cope with life in the mammalian host environment. So example one was an experiment that was funded by the New Investigator Award. And this experiment addressed what would happen if we removed a gene that was required for the fungus to sense and adapt to extracellular pH. So in the lab, the comparison between the parental isolate and the mutant isolate under acidic and alkaline conditions revealed that the deletion of the gene was unable to produce a fungus that could tolerate alkaline adaptive growth. And in the host, the deletion of this gene that was called PAX-C led to an inability of the hyphae to puncture the airway epithelium. And this stood in stark contrast to the invasion process of the parental isolate, which had dissolved the airway architecture after 72 hours of infection in mice. And at the level of whole animals, the pH non-sensing mutants were largely unable to produce a fatal disease in these animals, with 100% mortality in the mice infected with the mutant at day 6, compared to the parental isolates, where almost 80% of the infected animals had died. So these results told us that pH signalling is important for inhaled fungal spores to colonise the mammalian lung, and suggested that this signalling pathway could be a viable target for novel antifungal drugs. So the next example was a question that addressed the rewiring of the pH signalling pathway to shut down its response in the presence of drug. And so in this experiment, we wanted to establish a fungal lesion in the mammalian lung and then shut down pH signalling to see whether or not we could cure those lesions. And the drug that we used was called doxycycline. So again here, looking at the results of that experiment in the laboratory, what you'll appreciate is that in increasing concentrations of doxycycline at acidic or alkaline pH, the mutant was unable to adapt to alkaline conditions in the lab. And in a mouse experiment, the number of lesions in the infected lung was reduced in the presence of doxycycline by a factor of 60% by day 6. So this told us that there was genuine therapeutic potential associated with this pathway. But what it didn't tell us was how this effect was being elicited. So we asked, well, what happens in the lung? And we know that when fungal spores enter into the alveolar environment, they stick to the resident lung cells. And in the absence of an effective immune response, those spores swell and they generate hyphae, which in the absence of innate immune defences will germinate and generate these invasive hyphae, which have to adapt to the host environment to produce secreted enzymes and toxins and rupture the blood vessels to cause tissue damage. So in our next experiment, we addressed the total transcript repertoire of fungus growing in two different models of disease, a model of chronic aspergillosis and a model of invasive aspergillosis. And we asked, well, what's the difference between laboratory culture? And we performed this experiment with mice that were infected with both parental isolate and a mutant that lacked the transcription factor driving alkaline adaptation. So in this movie, we're now looking at transcript abundance across the entire genome of Aspergillus fumigatus, and we're looking at the time scale of infection from 4 to 72 hours. And in areas where we see these big red circles, we're seeing genes that have been activated and are being expressed at high levels. And in green circles, genes which are being reduced in their expression. And from scrutinising these data, we were able to identify that the transcription factor that governs alkaline adaptation and is critical for pathogenicity in the host was actually governing a programme of gene expression that drives all of the secreted products that the fungus requires during colonisation of the host. And this included the cell wall factors, the secreted proteases that are needed for tissue degradation, and cell wall biosynthesis. We also found, which was very interesting, that the conditions encountered by the fungus in both the steroid-treated animals and the acutely immunosuppressed animals was actually pretty similar. In all of our subsequent experiments, we were able to show that the pH non-signalling mutants were unable to cause damage to lung epithelial cells in the lab. They were unable to degrade the matrix that holds those lung cells together in the lung, unable to produce toxin that kills lung epithelial cells, and importantly, were more sensitive to drugs that target the fungal cell wall. So this work gave us confidence that what we were working on here was a pathway that would be critical in terms of governing virulence, and it gave us confidence to look at the next experiment, which was to rewire the fungal pH signalling pathway to promote alkaline growth when pH signalling is dysfunctional. And we did this by rerouting regulation of a transporter that was critical for utilisation of a nutrient required under nutrient stress. And that would only be expressed when the signalling pathway was switched off. And then instead of looking at genetic inhibitors of fungal pH signalling, we looked for chemical inhibitors. And we did this using a library of chemical fragments, 500 or so in our first experiment. And we looked for wells where the fungus was able to grow, which indicated that the signalling pathway had been turned off. And it gave us a very good signal to noise ratio and identified a number of molecules which act specifically on this signalling pathway. Now, working recently with medicinal chemists, we've been through several cycles of design, make and test and identified 7 off-the-shelf derivatives of one of these molecules that has extremely high potency in the nanomolar range. And through all of our genetic screens indicates that we're achieving on-target inhibition of fungal pH signalling. Moreover, the application of this agent to azole-susceptible fungal isolates in the presence of existing antifungal agents is synergistic, i.e. augments their activity. And this is also true in the case of azole-resistant isolates, where application of the novel agent overcomes that resistant phenotype. So these are very promising data indeed, and we now have BBSRC funding to follow those up. Now, we're fully aware of the major issues that we have ahead of us with this programme of research and the issues that are experienced in the later phases of drug design and discovery. But we've built a programme that is heavily reliant upon medicinal chemists and industry collaboration. And we will seek to work with our excellent clinical colleagues in the centre who have enormous expertise in the design of clinical trials and the application of PK/PD models of tissue distribution in animal hosts. So finally, just to round up the talk, I want to say that what I've spoken to you about so far is a genetic dissection of a single signalling pathway that's important for fungal pathogenicity. but actually we can apply these analyses at the genome scale. And in Manchester, in collaboration with Mike Bromley's group, that's exactly what we did. We approached the deletion of transcription factor genes in high throughput, and then we infected mice with pools of these mutants. Again, we used 2 models of mouse disease. We harvested the lungs and we looked for the mutants that were most fit at several days post-infection. We used a molecular method to detect the mutants which dropped out of the infection and were therefore less fit. That approach worked very well, and we've identified a cohort of 40 or so transcription factors in Aspergillus fumigatus, which impact fitness in both the heavily immunosuppressed animals and those which are treated with high-dose corticosteroids. And we've mapped those transcription factors back to the stresses that they support adaptation to. And we've demonstrated that many of these transcription factors that had been previously uncharacterised are important for fungal pathogenicity. So the hope is that having developed methods that can reroute these signalling pathways and drug discovery screens that can identify chemical inhibitors of them, we're at a point where we can quickly scale up, let's say, the repertoire or the preclinical medicine cabinet that we can then work harder on to find the next generations of agents that might be useful for antifungal therapy. And with that, I'd like to acknowledge a whole host of collaborators that I've worked with over the years on this project. Bill Neerman was highly instrumental during the early years of my programme in assisting the whole genome transcriptomic studies. Herb Arst is my collaborator on the inhibitor project. He was also my PhD supervisor at Imperial College. I want to recognise the entire team at the University of Manchester with whom we worked on the genome census project, and also the team who are working on the novel inhibitors of pH signalling, as well as my colleagues at the CMM, the Centre for Medical Mycology in Exeter, who have really helped me to settle in and re-establish my research programme here, but who are also incredibly enthused about the strategic relevance of looking for novel antifungal therapies. And we've made some strategic moves to appoint and to establish infrastructure in this area going forwards. And with that, I'd like to thank you for your attention.